Welcome to the fourth screencast on genetic engineering. This screencast looks at two other examples of genetic engineering: xenotransplantation and gene therapy. So dealing with xenotransplantation, firstly, xenotransplantation is the idea that we could use animal organs and animal tissues to transplant into humans. There is a tremendous shortage of transplant organs for the donor service, so things like kidneys and livers. Pigs, which are anatomically similar in size to humans, provide a possibility of being able to use, let's say, a pig kidney to transplant into a human being, which would give us a fairly limitless source of donor organs for the donor service. Okay. This isn't necessarily as easy as it sounds, however, because we have problems with tissue rejection. Okay, the problem being that pig cells have on their cell surfaces pig antigens, and those antigens would be recognized by the human recipient, and the B and T lymphocytes would create an immune response and would very quickly reject that transplanted organ. Okay, so what we could do is to try and genetically engineer pig cells to not express their pig antigens, and similarly we could also genetically engineer pig cells to express human antigens. Okay, so after a lot of genetic engineering, we could produce pigs that had cells which, in terms of their immunity or their antigen structure, would be very similar to human cells, which would reduce the chances of tissue rejection. Okay, so there's two things that happen here. First of all, we can insert human genes to cell surface proteins, okay, and similarly we can inactivate or get rid of the cells which would make the pig antigens. Okay, so if I try and represent all of that diagrammatically below, the first idea I'm going to deal with is trying to remove pig antigens, and this is sometimes called gene knockout. Okay, so here we have a pig cell. Okay, so we would take a pig cell, in theory any pig cell will do here, okay, and the pig cell has on its cell surface characteristic molecules, usually glycoproteins, okay, which are antigens, okay, and these are the ones which would be very, very different to human antigens and would be recognized by a human immune system, and we would very quickly make antibodies to this and killer T cells, etc., from your F215 course, which would very quickly reject any pig cells that we put into a human being. Okay, so what we try and do is that we work out that there's some form of process which involves a gene, okay, usually making enzymes, which is involved in inserting these glycoprotein molecules onto the cell surface. Okay, so we could find that gene and somehow disrupt it. Okay, we would disrupt that whole pathway, which would stop this cell being able to put pig antigens on its cell surface. Okay, so just going through this in a sort of bulleted point type situation, just to show you what would happen. I'm just reading from these. We would have pig cell antigens provide hurdle to xenotransplantation, as would elicit an immune response, as would be detected by T and B lymphocytes, as we just said. So we take a pig cell, and what we do is we insert a length of DNA into the middle of the gene which is responsible for pig antigen synthesis. Okay, so we've identified the gene, okay, and what we do is that there are specific restriction enzymes. In fact, they're called zinc finger nucleases, okay, although you don't need to know that, okay, which can specifically find a gene, okay, and cut the middle of it, cut into the middle of it. We then insert a random piece of DNA into the middle of that gene, which basically means that gene is not transcribed and translated into the same protein. Okay, so the protein would be non-functional, and if the protein was non-functional, the biochemical pathway which leads to antigens being inserted into the cell surface here would not be functional, and we wouldn't get pig antigens. Okay, so that's what happens there. Okay, so as I said, the pig cell is no longer capable of producing pig antigens. Okay, so now we've got a cell with a nucleus in it, and that cell doesn't produce pig antigens. What we then do is a bit of nuclear transfer, okay, which we'll talk about again when we do cloning, which is one of the next topics. But what we would do is we'd remove the nucleus from our engineered cell and insert it into an anucleated egg cell, a pig egg cell which had its nucleus removed, okay, which we call an anucleated egg cell. And we'd then stimulate that egg cell to divide to form a pig embryo. Okay, we would then take that pig embryo and we would insert that pig embryo into the uterus of a surrogate sow. Okay, and a few months later what would happen is a pig would be born, okay, and all of the cells in that pig would no longer or wouldn't have pig surface antigens. The idea being that those pig cells, if we did transplant them into humans, okay, what would happen is that they wouldn't elicit the immune response that normal pig cells would do. Okay, so here's a little diagram. Okay, so we've got our pig, we've taken some cells, okay, and these are the cells we're going to engineer, or one of these cells we're going to engineer. Okay, so it's this cell that we insert this piece of DNA into the middle of the gene responsible for making pig antigens. Okay, once we've transformed that cell, what we do is we take the nucleus out of that cell and insert it into an anucleated egg cell. Okay, stimulate that to divide, okay, into an embryo. We then insert the embryo into a surrogate pig, surrogate sow, and that develops into this genetically engineered pig. Okay, so none of the cells in this pig would have these pig antigens. Okay, so in theory you could harvest its organs, its kidneys, its heart here, all sorts of things, and use those to transplant into a human being, and this human being's immune system wouldn't have an immune response towards those pig cells. Okay, as well as knocking out pig genes. Okay, what genetic engineers have also done is they've knocked in, it's called gene knock-in. Okay, they've knocked in human genes. Okay, so in this case what they do is that they would take a gene for human cell antigens. Okay, so human cells produce characteristic antigens, okay, and there's a gene responsible for that. They isolate that gene and insert that gene into cells of a newly fertilized pig embryo. Okay, so you've got these genes which have been inserted into your pig embryo. Okay, and as that pig develops, it will eventually be born, okay, with expressing antigens which are human cell surface antigens on its cell surface. And again, this acts to reduce rejection if those cells were then transplanted into humans. Okay, so just going back to the main slide which you've got as part of your notes, just to make sure you're clear on everything. So you need to know what xenotransplantation is, okay, which is basically foreign, transplanting tissue from foreign or different species, okay, into humans. Okay, pigs are often the chosen ones simply because their organs are similar size to humans. Okay, problem, tissue rejection. Okay, get around this problem, genetically engineering the pig cells, not to have pig antigens, and also genetically engineering them to display human antigens. Okay, and then once you've engineered the cells, okay, you then do some cloning, okay, by nuclear transfer, cloning. Okay, take the cell, put it into an empty egg cell, make it divide into embryo, and then you have an animal which has, which doesn't show pig antigens, but can also display human antigens, and those cells or organs will be much more effective at being transplanted into a human. Okay, so that deals with xenotransplantation. The next slide looks at another application of genetic engineering. Okay, that of gene therapy. Gene therapy is an idea that offers potential treatments to sufferers of genetic disorders. You remember, even from GCSE, learning about genetic disorders such as cystic fibrosis, which, if you remember, was carried on a recessive allele, Huntington's chorea, which is carried on a dominant allele. If a disease such as cystic fibrosis provides particular problems in a particular organ, which cystic fibrosis causes particular problems in lung tissue for sufferers, what it could be possible to deliver a gene into the defective tissue, and that gene would then be transcribed and translated into a protein which return normal function to that particular tissue. Okay. So if the disease is carried by a recessive allele, what we would want to do is to insert a copy of the dominant allele into the cells. And if the gene was caused by a dominant allele, what we would need to do is to silence or knock out that dominant allele, and we could do this by inserting a piece of DNA into the middle of that dominant allele so that it cannot be expressed. Okay, so one of the things that we must do is to actually get these alleles, or these bits of DNA, into the middle of dominant alleles. We need to get those alleles into our tissue. Okay, we need to do this by vectors. Okay, so we've come across these words before. Viruses, plasmids, and liposomes would all be good vectors to get our gene, whether it's a copy of a dominant allele, into cells where there is two faulty recessive alleles, or if there is a cell which has a faulty dominant allele, by silencing that dominant allele by inserting a piece of DNA into the cell to disrupt the activity of that gene. Okay, so getting the gene into the cell, we would have to do via a vector. Okay, viruses, plasmids, or liposomes. Okay, I've written here something that's in the previous slide. A liposome is, if you like, an artificial phospholipid vesicle which allows DNA to get through membranes. Okay, so I've got an example here, which is often talked about in gene therapy, is an example of cystic fibrosis. Okay, so cystic fibrosis, if you remember, is a disease which is caused by having two recessive alleles, being homozygous recessive for the faulty cystic fibrosis allele. In terms of what it does, the faulty gene results in mucus being produced, which is rather thick and sticky and isn't cleared by cilia, and creates all sorts of lung problems for cystic fibrosis sufferers. Okay, so what we could do is get a dominant allele for the normal healthy allele, which produces normal non-sticky mucus, insert that allele into the vector, which in this case is a virus. Okay, and the virus is then inserted into the lung. Okay, this can be done with aerosols, much in the same way as you use an inhaler for asthma. Okay, an inhaler which would contain a suspension of these viruses which carry a normal dominant allele. Okay, those viruses then infect the cells lining the lung. Okay, so these are the cells which contain, well, like any of the cells, contain two faulty recessive alleles. Okay, and they insert a copy of the dominant healthy allele into the cells it comes into contact with, and the idea is that those cells will now produce normal non-sticky mucus which will alleviate the symptoms for the sufferer. Okay, so moving on to the next slide, to distinguish between two types of gene therapy. The gene therapy I've just described would be an example of somatic gene therapy. This is gene therapy when we are just inserting genes into normal body cells, okay, and altering the function of those body cells. Okay, one of the problems with gene therapy, okay, it isn't usually permanent, okay, because those genes aren't passed on in mitosis to other cells. Okay, so as the cell dies, okay, the problem returns. Okay, so we have to keep reapplying our therapy, our gene therapy, to the cells of the lung lining, also the cells that line the lung. Okay, so that we keep producing mucus which is non-sticky. Okay, and the altered genes can't be passed on to subsequent generations. So cystic fibrosis sufferers, okay, or even cystic fibrosis carriers still pass on the gene for cystic fibrosis in exactly the same way, even if they've had gene therapy. Okay, so that's somatic gene therapy. Okay, the other possibility is what's called germline gene therapy. Okay, this is when we insert genes, okay, or alter the genes, or silence genes or knockout genes in either sperm cells or egg cells, or in newly fertilized embryo cells, okay, or stem cells, okay, and the idea being that once you've genetically engineered these sperm, egg, embryo, stem cells, is that those will then produce individuals, okay, all of which, or the cells of which, will all contain the recombinant genes, okay, and the idea is that also those individuals will pass on those recombinant genes to the next generation. Okay, this is presently banned in humans. Okay, but in actual fact, we've looked at this with our pigs. Okay, so our knockout genes in pigs that we looked at when we looked at xenotransplantation is a bit like germline gene therapy, okay, for the pig. Okay, so to illustrate what the possibility of germline gene therapy would be, okay, so here I've got an egg cell, okay, this is a fertilized egg cell, okay, and this fertilized egg cell has a faulty gene, okay, for this example, this particular type of anemia, okay, the details of which aren't important here, okay, but there's a faulty gene, okay, in this particular case it's caused by two recessive alleles, okay, so what we do is we insert, okay, a dominant healthy allele into this fertilized egg cell, okay. These fertilized egg cells then divide by mitosis, okay, to produce an embryo, and that embryo eventually develops into 9 months later into a child, okay, and this child will not be a sufferer of the disease caused by these two recessive alleles, because all of his cells contain a dominant allele, a dominant healthy allele. Okay, so that's an example of germline therapy. And as I say, there's tremendous concerns about germline therapy in human beings, so it is banned, or particularly in most Western countries, it would be banned. Okay, so moving on to the last this unit, we look at ethical concerns raised by genetic manipulation. Okay, so there's often questions where you have to list ethical concerns or worries about genetic engineering technologies. Okay, so just going through some of these. Okay, I've got no diagrams for this particular slide. Okay, but just going through these. Okay, so in terms of genetically engineering microorganisms, one of the key concerns is, as you know, we used antibiotic marker genes. Okay, we transferred antibiotic marker genes from one organism to another. Okay, the problem with antibiotic marker genes is that these antibiotic marker genes in plasmids, okay, could be passed on, okay, through conjugation tubes to pathogenic bacteria. Okay, so those pathogenic bacteria would now be resistant to antibiotics. Okay, and if those pathogenic bacteria became widespread in the population, we might not have any antibiotics to be able to control the diseases caused by these pathogenic bacteria. Okay, and that could be a real problem. Okay, in terms of plants, what are the issues with plants? One of the idea is resistance for herbicides. Okay, so we've genetically engineered plants to be resistant to herbicides, so that our crop plant is resistant to herbicide. We then spray the herbicide over the crop, kills all the weeds, but the crop plant survives. Okay, but if those genes for herbicide pass into weeds, okay, suddenly we might have a population of superweeds which we can't control with normal herbicides. Okay, and that would create a huge problem for agriculture and ultimately feeding the world population. Okay, similarly, pest resistance. Okay, we can engineer plants to be resistant to pests. Okay, such as insects. Okay, if use of pest-resistant genes in crops becomes so widespread, okay, what would happen is you could do such an effective job of killing off insects that you would completely collapse food chains. Okay, so no insects equals no birds, and that food chains and food webs in particular areas would be dramatically altered, altering the ecology of the area. Okay, and also genetically modified crops. Okay, GM crops. There's an awful lot of stuff in the press about GM crops. We're very happy to eat GM crops, but we don't really know what the long-term health implications on humans might be. These haven't been around that many years. It's possible that in 20 or 30 years' time, there might be a whole host of diseases such as cancers and so on, which we didn't realise was going to be an implication of of humans eating genetically modified crops. Okay, the other idea is that if you have engineer crops to be resistant to disease, what happens is that the pathogens, what they do is they evolve. Okay, and you could end up causing the production of some really devastating pathogens, which are very difficult to counter by genetically engineering disease resistance into crops. Okay, in terms of genetic engineering of animals, okay, there's obviously concerns about animal welfare. In terms of obviously the for example, when we talked about genetically engineered pigs for xenotransplantation, okay, there's obviously religious issues about various religious groups would not be able to partake of transplant organs from pigs, so that would be a religious issue, and obviously concerns about animal welfare, testing of genetic engineering on animals, etc. Okay. And in terms of humans, again, we're not very sure what the side effects of genetic gene therapy and genetic engineering in humans might be, and there's also would be concern of germline gene therapy to produce cosmetic traits in unborn children, which would be ethically unacceptable. Okay, so those are just a series of ethical concerns relating to microorganisms, plants, animals, and humans. Okay, so that finishes off our look at xenotransplantation, gene therapy, and ethical concerns arising from those technologies.